Okay, so finally we are going to start the very final piece of our final chapter, which is the male reproductive system. So the primary reproductive organ for the males are going to be the testes, because that's the gonads. And we've also got accessory organs and glands. We've got a very complex set of ducts and tubules that are going to go from the testes all the way to the penis, and that's how we're going to get our sperm from the testes through the penis. And then we've got a whole bunch of accessory glands, and then you've got the penis itself, which of course is the organ of copulation. So looking at the scrotum for starters, the scrotum is a sac of skin located between the thighs. It provides an environment about 3 degrees lower than body temperature. So our sperm produce more efficiently, more effectively at roughly 3 degrees lower than body temperature. And so putting the testes, which is where the sperm are going to be produced, in the scrotum kind of keeps them away from that core body temperature, and it helps to provide sort of a cooler environment for them so that we can produce them more efficiently. The scrotum wall contains a smooth muscle layer just inside of the skin called the dartos muscle. The dartos muscle can contract and relax in response to changes in temperature. So if you can imagine that the testes maybe are exposed to a colder environment, that dartos muscle can contract and it pulls the testes closer to the body, so it lifts them closer to that core body temperature to warm them up, to keep them closer to that ideal temperature. Conversely, if we're exposed to like a hotter or warmer temperature, that dartos muscle would relax and then cause them to descend further away from that core body temperature, providing them, in that case, a cooler environment. So the testes are located within the scrotum basically because we need a little bit cooler temperature than body temperature for ideal sperm production. And then we've got that dartos muscle just inside the skin that can contract and relax to kind of bring them closer to the body if we need more warmth, and further away from the body if they need less warmth. The testes are paired oval organs, again, that are housed within the scrotum. The function of the testes is going to be to produce sperm and androgens. So androgens is a term that we've used before, and I told y'all that it's kind of the collective term that we use for testosterone and other molecules that are structurally and functionally similar to testosterone. They all fit into the category of androgens. So if you look at here's the testis here, and there's some layers that cover it. This white area immediately touching the testis is called the tunica albuginea. And then you see this layer right here going alongside. This is called the visceral layer of the tunica vaginalis. And then this outer layer here is called the parietal layer of the tunica vaginalis. So just a couple of kind of membrane structures that are wrapping around the testes. Now the tunica albuginea, so this is this whitish area that covers the testes. This has these inward projections. You can follow one there. Here's another projection there. There's another projection there. Those are called septum or septa as it projects in. And it divides the inside space of the testes into these different lobules. So if here's a septum, and here's a septum, you've created this area in here. This is called a lobule, and we've got about 250 separate lobules. Each lobule contains up to 4 convoluted, thin, seminiferous tubules. So these ramen noodle looking things in here, you may never eat ramen noodles again, but what that is, those are the seminiferous tubules. So they're very thin and convoluted because they're kind of twisty and curvy. And that's where sperm production is going to take place, inside those seminiferous tubules. So the seminiferous tubules contain 2 types of cells. One of them is a group of non-dividing support cells. These are called sustentacular cells. These are also sometimes called Sertoli cells. And then you've got a population of dividing germ cells. So germ cells meaning that when you hear the term germ cell or germline, that means somewhere in the future it's going to be responsible for producing like a gamete type of cell. So we've got a dividing germ cell line that's going to continuously start to produce the sperm once this individual reaches puberty. So the function of the sustentacular cells are to provide a protective and a nourishing environment for the sperm. So because sperm are going to be made right here in this environment, the sustentacular cells are there to kind of give them what they need. And then to release the hormone inhibin. So this should ring a bell. We talked about inhibin in the female reproductive system. Inhibin is going to inhibit FSH secretion, and so it's going to regulate sperm production, because we're going to see how FSH affects sperm production here in a bit. But you should recognize inhibin, and just like it did in the female reproductive system, inhibin is going to inhibit FSH release from the anterior pituitary. Okay, so whenever sperm levels get really high, we'll start to release inhibin, and inhibin will inhibit FSH, and that helps to kind of slow down the production of sperm. Conversely, when sperm production decreases, inhibin secretion decreases, and that allows us to produce more FSH to kind of ramp up sperm production. So interstitial cells, also called Leydig cells, are found in between the seminiferous tubules. So here's a seminiferous tubule, here's a seminiferous tubule. So you have cells that lie in between there that are called Leydig cells, again, or interstitial cells. So these interstitial cells respond to luteinizing hormone. Luteinizing hormone stimulates these interstitial cells to produce those androgens, or testosterone. So we do get some amount of androgens being produced by the adrenal cortex, but specifically in a male, the vast majority of androgen synthesis and release is going to come from these interstitial cells in the testes starting at puberty. Here we're going to look at how the hormones, and these are going to be the same hormones that we saw in the female reproductive system, how they regulate androgen production and sperm development, which is called spermatogenesis. So you have the hypothalamus that starts the whole process of spermatogenesis by releasing gonadotropin-releasing hormone, or GnRH. GnRH, we know, goes and communicates with the anterior pituitary to cause the release of FSH and LH. So FSH and LH, these in both sexes, these target the gonads. In the females, they head down to the ovaries. In the males, they're going to head down to the testes. Specifically, luteinizing hormone is going to stimulate those interstitial cells, or the Leydig cells, to secrete testosterone. And the FSH is going to stimulate the sustentacular cells to secrete androgen binding protein, or ABP. Androgen binding protein binds to androgens, and it essentially allows a mechanism to keep the hormone levels very concentrated there in the seminiferous tubule, so we have a very high testosterone level there. So the high testosterone level is going to have a variety of different effects on the body. It's going to facilitate spermatogenesis there in the seminiferous tubules. But just like we saw in the female reproductive system, testosterone is also going to have kind of this negative feedback role when it comes to the brain. So it's going to inhibit gonadotropin-releasing hormone secretion, and then it reduces the anterior pituitary sensitivity to GnRH. So rising levels of testosterone, again, are having a negative feedback on that entire cycle. So the GnRH leads to increased luteinizing hormone, and FSH, those are going to lead to higher levels of testosterone. And as we get higher and higher levels of testosterone, it's going to negatively feed back on the brain, so we'll get lower and lower levels of GnRH, FSH, and LH. Now, the sustentacular cells are going to respond to that rising sperm count and start to release inhibin. So inhibin primarily causes the inhibition of FSH secretion from the anterior pituitary. We saw that in the females as well. And it's going to serve as an additional negative feedback mechanism. So the higher levels of testosterone are negatively feeding back to the brain to prevent more and more FSH from coming out. but the sustentacular cells also are going to start to release inhibin in response to rising sperm counts. So that inhibin can then go inhibit FSH. Okay, so circulating testosterone throughout the body is going to, so before I talked about high testosterone in the testes and that promotes sperm development, but as it circulates through the body, it has a variety of responses as well. It stimulates libido. It stimulates the development of secondary sexual characteristics, hair growth, deeper voice, larger muscle mass, things like that. Testosterone can also act on the brain, so there's an increased desire and sensitivity to sexual stimulation. So here is our lovely picture where you can see all of that. So we'll start in the hypothalamus. GnRH is getting released. It's talking to the anterior pituitary, which is then going to release FSH and LH. So LH is going to go to those interstitial cells or the Leydig cells, and it's going to stimulate the release of testosterone. So here is the interstitial cells releasing testosterone. Testosterone is going to do a couple of things. One of the things it's going to do is going to stimulate spermatogenesis. The other thing, it's going to have an inhibitory or a negative feedback mechanism on the brain. So we reduce GnRH, and we reduce the hormones coming from the anterior pituitary, which is FSH and LH. Okay, so the FSH is going to stimulate those sustentacular cells to release androgen binding protein. So here's the sustentacular cells. They're going to release androgen binding protein, which is going to bind to the testosterone, and it's going to keep the testosterone levels high here in the seminiferous tubules and in the testes. So the testosterone stimulated spermatogenesis. So spermatogenesis, we're going to get rising sperm counts, and that is going to lead to the release of inhibin. And I'm trying to find inhibin on this. Oh, here it is. Okay, so rising sperm count, we lead to inhibin, and here comes inhibin, and it, we already know from the female reproductive system, feeds back and has negative feedback on the brain. So it tells GnRH to stop, it tells FSH and LH to stop. Okay? And then this last little bit right here, testosterone stimulates libido and the development of secondary sexual characteristics. Spermatogenesis is a process of sperm development, and it's going to happen in those seminiferous tubules within the testes. Of course, it's only going to begin at puberty once those levels of FSH and LH start to be released from the anterior pituitary. All of the sperm develop from this primordial germ cell called a spermatogonium. Spermatogonium are diploid cells. They have 23 pairs of chromosomes, so 46 total, and they lie near the base of the seminiferous tubule surrounded by the cytoplasm of a sustentacular cell. So what you're looking at here, this is inside a seminiferous tubule, where this right here is sort of the outer edge of the tubule. This beige or pink is the lumen or open space of the tubule, and this is the cells that are lining on that outside base. So you can see the spermatogonium is right here on the very outer edge of the seminiferous tubule. So the first thing that has to happen is the spermatogonium have to replicate, or divide, I should say, by mitosis. If we take the spermatogonium and put it directly into meiosis, we're going to run out of spermatogonium. And spermatogonium are what we need to make the sperm. So the very first thing that has to happen is we have to make a copy of the spermatogonium. So the first thing it does is undergoes mitosis, and that produces what we call two primary spermatocytes. Well, typically one of these stays a spermatogonium and the other one becomes a primary spermatocyte. So the primary spermatocyte is also a diploid cell, 46 chromosome, but that's the one that's going to enter into meiosis, and you can see it becoming kind of meiosis 1 here, and then those are going to go through meiosis 2 until eventually we're going to create our sperm in the seminiferous tubule. So when the primary spermatocyte, so here's our primary spermatocytes, they're going to go through the process of meiosis 1. So they go through meiosis 1, and it produces two cells. These are called secondary spermatocytes. The secondary spermatocytes are haploid cells, so they only have 23 chromosomes each. They're still surrounded by that sustentacular cell cytoplasm, but you can see that they have moved closer to the lumen. So essentially what's happening is you start with your spermatogonium. You're going to go through this whole process of mitosis and then meiosis 1 and then meiosis 2. And as the process is happening, they're moving closer and closer to the lumen of the seminiferous tubule where they'll just kind of fall in. Okay, so secondary spermatocyte, I mentioned that they are haploid cells. They have 23 chromosomes. They're still not genetically where we need them to be in order to become sperm, because at this point we have 23 replicated chromosomes. So yes, it's haploid because there's only 23 chromosomes, but each one of those chromosomes is replicated, and we can't have that. So that's why we have to undergo meiosis 2 to get them to 23 single chromosomes. So the secondary spermatocytes have to undergo meiosis 2 to become spermatids. Okay, now spermatids are also haploid, but these contain 23 single chromosomes. So these are of the proper genetic makeup to be a proper gamete. Okay, the only thing missing here is that they're the wrong shape. These are still kind of a circular type shape. So all they have to do really is change their morphology or change their shape to become, the typical head-tail structure that we know that sperm have. Spermiogenesis is the final stage in spermatogenesis. So notice that I've changed the name up here. We were talking about spermatogenesis. The spermiogenesis just refers to the final stage of spermatogenesis, and that's when those newly formed spermatids differentiate. And what that means is they become anatomically mature, meaning they basically evolve from this circular shape to the head-tail structure that we are all familiar with for sperm. So during spermiogenesis, you get a structure called an acrosome cap that forms over the nucleus. It's going to be kind of in the head region. The acrosome cap contains digestive enzymes, and those are going to be important so that when that sperm encounters an oocyte, those digestive enzymes will help to penetrate through the protective barrier of the oocyte so that the nuclei can join together. As the spermatate elongates, you get the formation of a tail or a flagellum that's attached to the midpiece. So this would be kind of the head of the sperm. This would be the midpiece region, and then extending from that is the flagella. The neck region, which is right here, this contains a lot of mitochondria and a centriole. The mitochondria are essential because these sperm are going to need a lot of energy because they have a long journey ahead of them, so they have lots of mitochondria there to help move the tail. Okay, so here we have the sperm. They have the proper genetic makeup. They have the proper anatomy, but they're still not fully mature in that they're not motile at this point. So from the inside of the seminiferous tubules, they'll have to go to a structure called the epididymis, and they kind of hang out and spend some time there while they gain their motility, and then they will be fully functioning sperm. This slide I really just kind of put in there again so you can kind of see the process from spermatogonium to primary spermatocyte, meiosis 1, secondary spermatocyte, meiosis 2, spermatids, and then they'll go through spermiogenesis, and then they get their structure. And then I wanted to give you this kind of up-close image right here so you can see the head of the sperm, which contains the acrosome cap. You can see the bottom of the nucleus there, the mid-piece or the neck region, that's where you're going to have a whole bunch of mitochondria, and then you have the tail or the flagellum extending from that. Okay, so spermatogonium, primary spermatocyte, secondary spermatocyte, spermatid, and then spermatozoa, which is the mature sperm. So just kind of different terminologies for the process of creating the sperm. This is their chromosomal makeup. This is whether they're diploid or haploid, and then kind of where they are in the process. So each testis has a set of ducts to store and transport the sperm as they mature and pass out of the body. The reedy testes is a meshwork of interconnected channels that receives the sperm from the seminiferous tubules. And then the channels of the reedy testes merge to form what are called the efferent ductules that drain into the epididymis. Okay, so here are the seminiferous tubules. They're all in those lobules. And then here are the reedy testes. So the sperm will drain from the seminiferous tubules to the reedy testes. And then from there, it goes through these efferent ductules, which sometimes you see these called straight vessels, and you can see why they're pretty straight, and they go straight to the structure called the epididymis. This is the epididymis right here. Okay, so this is the structure where the sperm are going to spend some time while they kind of gain their motility. So seminiferous tubules, this is where spermatogenesis happens, and they go into the reedy testes, and then they go through the efferent ductules to the epididymis. So here you can see the epididymis again, right here. It's lying kind of outside the testes. It's a comma-shaped structure. It's got that internal duct system that we saw in the previous slide, and then it's covered externally by connective tissue. So inside the epididymis has this long convoluted duct called the duct of the epididymis. And again, that's where the sperm are going to be stored until they're fully mature and capable of being motile. So if they get expelled too soon, if they haven't spent enough time in the epididymis and they end up getting expelled too soon, then they aren't yet fully motile and they don't have the ability to swim, so they won't be able to travel through the female reproductive tract, and ultimately they won't be able to reach the oocyte. If they're not ejected in a timely manner, if they spend too much time in the epididymis and they don't get expelled, then they basically degenerate and get reabsorbed, and then we'll replenish them with sort of newer sperm. The ductus deferens, or the vas deferens, is the very next place that the sperm are going to go on their journey. So it connects the epididymis to something called the ejaculatory duct. So this long tube is the ductus deferens, or vas deferens. So you see it goes all the way up. It runs underneath the ureter. So here's the ureter. The ductus deferens run underneath the ureter. As it gets close to the superior posterior edge of the prostate gland, so here is the prostate gland right here. As it gets close, it kind of enlarges. See how it's a very thin tube here? But as we get close to the prostate gland, it kind of enlarges. This is called the ampulla. It's the ampulla of the ductus deferens. The ampulla of the ductus deferens unites with something called the seminal vesicle, which is this structure right here. And you can see that right here, they're kind of opening into the same place. That's called the ejaculatory duct. Okay, so our sperm went from the seminiferous tubules to the reedy testes to the efferent vessels, or efferent ductules, to the epididymis. They spent some time in the epididymis. Then they're going to go through the ductus deferens, all the way up here, through the ampulla of the ductus deferens, which opens into the ejaculatory duct with the contents of the seminal vesicle. And you can see that's dumping right into the prostate. So the ejaculatory duct is going to conduct the sperm, which is coming from the ductus deferens, and a component of seminal fluid that's going to come from the seminal vesicle, and it's going to empty it into the urethra. So here's our urethra. Remember, we had the prostatic urethra, we had the membranous urethra, and then we had the spongy urethra. We talked about that in the urinary system. So the ejaculatory duct is going to empty right into the prostatic urethra, and then the urethra is going to transport the semen eventually outside of the body. So some accessory glands of the male reproductive tract, the seminal vesicles, we saw those. The seminal vesicles will secrete this viscous, whitish, yellowish alkaline fluid that contains both fructose and prostaglandins. Fructose is there to nourish the sperm, so they're basically going to take a really long journey and they're not taking much with them. So we send them out in the seminal fluid that has a bunch of fructose, so they kind of have the nourishment that they need as they go along. The prostaglandins are there to promote a very slight dilation of the external os of the cervix. So remember I said that it's mostly closed. I said that in the female reproductive system. It does open a little bit at the time of ovulation to help promote sperm entry, but seminal fluid has prostaglandin in there, and that helps to open the external os just a bit to help facilitate the entry of the sperm into the uterus. The prostate gland secretes a fluid that is weakly acidic. It's rich in citric acid and seminoplasmin, and also something called prostate-specific antigen, or PSA. So the citric acid is a nutrient for sperm health. The seminoplasmin is an antibiotic that combats urinary tract infections for the males. And then the PSA acts as an enzyme to help liquefy the semen following ejaculation. The bulbourethral glands, we've got 2 of those. Each one of them has a very short duct that projects into what's called the bulb of the penis, and then it ultimately empties into the spongy urethra. The product of the bulbourethral gland is this thin, clear, viscous mucin, which of course produces a mucus. The mucus then serves to kind of protect the urethra, and then also serves as a lubricant during intercourse. Now this last one I put in here just to kind of clarify the definition. This is not like a specific secretion from a specific duct, but when you use the term semen, what you're talking about is the contribution from all of these different glands in addition to the sperm that are coming out of the ductus deferens. So as you kind of follow the male reproductive tract, you get secretions from the prostate gland, secretions from the bulbourethral gland, etc., and then you mix that with the sperm that's coming from the ductus deferens to create what we know as semen. Typically, semen contains anywhere between 200 and 500 million sperm. Clinical view looking at the differences on benign prostatic hyperplasia and prostate cancer. So these both can affect the prostate. Benign prostatic hyperplasia, or BPH, is a non-cancerous enlargement of the prostate gland. It's basically kind of an overgrowth of the prostate. This tends to happen in older men. It has to do with hormonal changes associated with aging. The biggest complaint with benign prostatic hyperplasia is that it can make urination a little bit difficult. If you go back and look at kind of the anatomy, you can see that the prostatic urethra is running directly through that prostate gland. So if that prostate gland is swollen or enlarged, it impinges upon the space of that prostatic urethra, and that can make urinating quite difficult. Otherwise, BPH isn't something to be severely concerned about, as prostate cancer would be. Prostate cancer, of course, is an actual malignancy or an actual cancer. Risk factors, primary risk factor is going to be increasing age. So this is something that would have to be resected away or treated, of course, to make sure it doesn't get out of hand. Prostate-specific antigen is something that they look at in older men, and it can actually be indicative of either benign prostatic hyperplasia or prostate cancer. So if it's elevated, then they kind of have to go in and get a better look at what's going on. So I kind of ran through the gist of it. I do want you to run through and look. It talks about some of the procedures that you can do. So I want you to kind of read through and learn about the TERT procedure, understand about PSA, and that it can kind of be indicative of either benign prostatic hyperplasia or prostate cancer. The penis and the scrotum are what make up the external genitalia in males. So internally, the attached portion of the penis is known as the root of the penis. So you can see that here. So it's kind of where it's inserted on the inside of the body. And then you've got the body of the shaft of the penis is the elongated portion, and then the tip of the penis is called the glans. And that's where you're going to find your external urethral orifice. At the distal end of the penis, you've got the skin attached to the raised edge, and it kind of forms over the glands of the penis, and that's called the prepuce, the foreskin. So you can see that in here. It's not in this image, but you can see that over here. If you took a cross-section of the penis, you've got different erectile bodies. So you've got two corpus cavernosums. There's one. There's one. This is the primary erectile tissue. The corpus spongiosum is here. This is also erectile tissue, but it's kind of considered sort of a secondary erectile tissue. This is the one that contains the urethra. So the urethra runs through the corpus spongiosum, not the two corpus cavernosum. Okay, so each of the corpus cavernosum terminates in the shaft of the penis. The corpus cavernosum do not go all the way down to the glands of the penis, but the corpus spongiosum does. The corpus spongiosum goes all the way down to the glands of the penis. Another clinical view, so again, I'm not going to read through everything. Circumcision is the surgical removal of the prepuce of the foreskin of the penis. It's practiced among certain cultures. It's not practiced among every culture. It's got some health benefits to it, so I am going to again let you read through. It just talks about some of the benefits of it. This bottom talks about some of the drawbacks of it, so again, just familiarize yourself with circumcision. And lastly, I'll leave you with this clinical view. Again, not really going to go through the whole thing, but I do want you to kind of run through it. Sexually transmitted infections, or STIs, is a group of infectious diseases that can be transmitted through sexual contact. Keep in mind that does not always mean intercourse. It means any sort of sexual contact. Condoms have been shown to prevent the spread of STIs, but they're not 100% effective. A lot of times they're not 100% effective because they're not used properly, but even when they are, they're not perfect. So if I were to pick out one important thing that I would ask on a test that I would write, except unfortunately I don't get to write the exam for this one, I would want to kind of point out that genital herpes is the only one that's discussed on this slide that's actually caused by a virus. The other ones, chlamydia, gonorrhea, syphilis, these are all caused by bacteria, which means that while they're not great to have, they can be treated with antibiotics, whereas herpes is a viral infection. So just kind of read through, know the names of them, and which ones are caused by bacteria, which ones are caused by viruses, and things like that.